Hello everyone, my name is Zhengyang Guo. I am an undergraduate student from Dalhousie University. I'm glad to be invited to join the 5th International Conference Biology, Engineering, and Medical Science. My research is about CRISPR-Cas9 applications in T-cell modifications for tumor immunotherapy. Today, I will talk about the background of CRISPR-Cas9 and tumor immunotherapy. Also, I will introduce the theory of CRISPR-Cas9 T-cell modification directions, challenges, improvements, and conclusion. First of all, cancer is a serious and urgent health issue, which was considered as the second major case of death in the United States. The conventional treatment of tumors such as surgery, radiotherapy, chemotherapy, and hormone therapy have many limitations. However, CRISPR-Cas9 as a cancer immunotherapy powerful tool applied to the chimeric antigen receptor T cells, for short CAR T cells, and adaptive T cell therapy, for short ACT, and they generate an advantage in long-term specific target and the prokaryotic immune defense system for against foreign DNA invasion. They involved in foreign DNA integrate into the CRISPR array, then the infected CRISPR via transcription, cleavage, and match with activating RNA, forming guide RNA. Later, combine and active CRISPR-associated protein, for short Cas protein. Therefore, activated Cas protein able to recognize the PAM sequence and operate the process named non-homologous join, for short NHEJ, and non-homologous directed repair, for short HDR, to knock out genes, insert or correct genes in the absence or presence of donor DNA. TCR T cells directly bound target via major histocompatibility complex (MHC) in the variable region of TCR alpha and beta chain. Nonetheless, TCR CD3 complex formation is necessary for intracellular signaling components to activate T cells. The complete activation of T cells is required the signal 1 from MHC and signal 2 from co-stimulation of receptor regions paired intact with ITAMs in TCR3 complex 4. Therefore, TCR T cell therapy is performed by introducing CRISPR-Cas9 engineered TCR chains from new recognizing target incomplete TCR CD3 complex and removing endogenous TCR chains for avoiding mispairing among the endogenous chains with TCR chains. The CAR is designed for direct targeting of target surface antigens to activate T cells. The CAR contains an antigen binding domain from an antibody, a steric hindrance overcoming structure hinge domain, a cancer stability and an infection level related structure transmembrane domain, and one or more intercellular signaling domains. There are 3 generations of CARs. The first generation adapted CD3 zeta or FCR gamma signaling domain to activate T cells, but the effect did not reach the expectations. The second generation is the component of a co-stimulatory domain with the intercellular signaling domain of CD3 zeta. The third generation uses 2 co-stimulatory domains with the CD3 intercellular signaling domain. It inhibits improved function of higher synthesis of cytokine in lymphoma and pulmonary metastasis than the second generation, but did not present higher chemical effects in some models. The immune checkpoints, for short ICs, are the normal physiological immune system regulators located on the surface of immune cells. They have the ability of checking and balancing mechanisms to avoid incorporate immune response in host cells. PD-1 as a co-inhibitory receptor for PD-L1, which is expressed by the activated T cells. PD-1 targeted lesion PD-L1 is present on the surface of antigen-presenting cells, tumor cells, and normal tissue cells. Engage in trans interactions with PD-L1 to release inhibitory signaling on the surface of T cells to suppress T cell activities. Research showed knockout of the PD-1 gene in primary T cells and analyze the effect in M1 for melanoma cells and HSEBV gastric cancer cells from that found that the T cells have a consistent activation state and memory under the condition of a lack of PD-1 expression in increasing IFN gamma synthesis and tumor lysis ability. Also, CTLA for knockout T cells have enhanced anti-tumor ability on HCT116 colon cancer cell line, in weakening the tumor cell activation, increase late apoptosis tumor cells, and in promoting cytokine expressions. Harris research gathered statistics about CRISPR-Cas9 application in enhancing T cells cytokine synthesis by editing T cell genes. Editing some gene exits to cytokine secretion changes. Interferon gamma, for short IFN gamma, is mainly produced by activated NK cells and T helper 1 cells. It with IL-1/2 are able to co-induce the transcription factor T expression. Also, the inducing cause stimulation of IFN gamma expression. Furthermore, the type 1 interference can induce INF gamma expression via STAT4 mediated signaling in NK cells. However, it regulates many immune cells. For example, in macrophages, it stimulates pro-inflammatory cytokines expression and downregulates the synthesis of anti-infectionary cytokine. And it also enhances the function of eccentric cells, T cells, T follicular helper cells, and B cells. TNF-alpha is multi-purpose pro-inflammatory cytokine that can control the wide range of processes to inflammation, vascular functions, insulin sensitivity, cellular apoptosis, tumor growth, and invasions. It with its receptor able to enhance lymphoangiogenesis. It also stimulates TAMs promote lymphatic vessels within the tumor and increase lymph node metastasis. And the double knockout in enzymes diacylglycerol-kinases in human T cells improve INF gamma and IL-2 secretions and enhance T cell cytotoxicity in vitro and increased IFN gamma and GZB productions result in glioblastoma expression in the viral. However, they must have some challenges, and the common challenges for the TCR and T cells, including some types of tumor cells may escape from the engineered T cell by losing of the target antigen during long-term treatment. Second, the off-target toxicity feature in engineered T cells targeting and function on the normal cells. Third, the inhibitory factors in tumor microenvironment result in decreasing TCR and CAR T cell functions. And they also have some unique drawbacks of TCR are the downregulation of MHC in some tumor cells cause TCR lose efficiency, and also the heterodimer formations of exogenous and endogenous TCR result in new autoreactivity generation and graft versus host disease. And the unique challenges for CAR are some house cells possibly have target antigen that leads to target-induced tumor regression toxicity, and also maybe high affinity SCFV may cause off-target activation, while poor chemokine barrier coordination limits cell migration of infiltration. And they also have some improved methods for counter those problems. First, multiple targets designed T cell may able to against antigen escape. Second, cell gene editing strategy able to improve T cell availabilities. Third, cytokine regulation for improving T cell survival and function. Finally, CRISPR-Cas9 is a powerful gene editing tool which able to perform gene knockout and gene designing to treat tumors. This tool enabled CAR immunotherapy, which able to directly target cell surface antigens to induce T cell immune response, and TCR immunotherapy, which indirectly bound through MHC to activate T cells. Furthermore, CRISPR-Cas9 able to knock out immune checkpoints to enhance T cell function to prevent tumor escape, and also can be enhanced by modification gene in cytokine expressions. However, they also have some common and unique disadvantages, such as unexpected tumor escapes, off-target toxicity, downregulation of MHC in some tumor cells for TCR, and target-induced tumor regression toxicity for CAR. Nevertheless, some improvement methods can be used. For example, multiple target design T cell for counter antigen escape. However, my research only covered one of the concepts in tumor immunotherapy, and more potential methods can be adapted. Therefore, the other potential T cell identification mechanisms, immune checkpoints, and gene seeds in enhancing T cell functions and immune cells could be found focused on the further research of CRISPR-Cas9 application in tumor immunotherapy. And that's all. Thanks for listening. For more detail, you can reach to my research paper or connect me. And there are some references that I used in this PowerPoint.